So that makes things somewhat complicated in the mainstream medical community. This is a slide that's a little bit complicated, but it's actually really important. This is what basically makes large clinical drug trials in the US possible. And the success of each step is what drives the next step. So post-discovery and conducting of some preclinical studies, often done at universities, you need to obtain a strong patent, which will give you market exclusivity. Based on that, you can go and raise the financing you need to conduct the clinical trials. Then, as many of the doctors, as all the doctors here know, you get into phase 1, phase 2, and if those all successfully work out, you get into a very large clinical phase 3 study, which is hundreds or thousands or tens of thousands of patients. Obviously, we do have orphan drugs or some rare cancers where the FDA will allow you to have much smaller studies, but those tend to be smaller. You get the FDA approval, you establish pricing, and then you get medical insurance reimbursement. You're then able to inform the market and market what the DCs you're obviously targeting. And then finally, you can generate some profits to reward the investors that originally took the risk. So without this cascade and without strong patents and without this reimbursement mechanism, investors simply won't fund large trials. And a lot of doctors just have a hard time appreciating. They just say, take it, take it, get it IND, take it to the FDA, but it doesn't quite work that easily. This is just an article that highlights the fact that it's very difficult to get patent protection for natural products. So there is actually a relatively strong patent portfolio on AHCC, but it's just not strong enough to give investors the confidence to invest the hundreds of millions of dollars you need in order to take it to drug trials. So as a result, you kind of have to take this cascade and realize all of the things that are now basically disrupted. You get some patent protection, but not all. You can really raise much less money. You can do small phase 2 trials, but phase 3 trials really not viable. You can't go to the FDA for that reason. And as I'll discuss, there's very little regulatory pathway. You can't get reimbursement, which means it's all out of pocket for patients. You can't actually talk about what the compound does. And obviously, as a result, you can reward the investors. Now, there's some exceptions. There's some fish oil drugs you may be aware about, and they've been successful because they were able to define some of the actives in fish oil. And there's some large trials on nutritionals. So Mars, for example, funded a study on multivitamins and cocoflavanols. For those of you who are curious, both groups showed a benefit for cardio health, and only the multivitamin, interesting enough, actually showed a cognitive health benefit in quite a large study. But that's obviously a side issue. So to me, this is really a market failure, which is more of a term used in economics, but that's what really blocks patient access. I mean, imagine if of all these drugs that were able to become drugs, what if they couldn't, what if the actives could not be isolated, synthesized, and strongly patented? You would basically have no funding, no trials, no patient access, and people wouldn't get a benefit, so people would suffer. So the solution is really simple, theoretically, which is you just have to establish a regulatory pathway for nutritional products. FDA realized that was the case, so they established something called botanical drug license. But the only thing they really said is, well, we'll accept the non-simple compounds. You could have complex compounds. But they didn't really change the IND process or solve the IND issues. As a result, only two drugs were approved. And most nutritionals either have to be dietary supplements, a few of them can be medical foods, but the challenges remain. So then, companies are left to kind of DIY things. And you sort of try to get the best patent possible. You have to offer the product as a nutritional supplement. You can talk about immune support, cervical health. You cannot mention HPV in any way. You go out of your way to try to do some extensive safety work. And actually, Judith did both phase 1 and phase 2 metabolism to look at drug-to-drug interaction, tell people, look, it's safe. So even though you may not have 1,000 people in a trial, at least you can be confident in the safety. And then beyond that, you try to educate mainstream doctors so they understand the challenges and start recommending the products. You try to price low so people can afford it. You invest in third-party quality certification to give doctors confidence. You can explore the medical food angle, which is something we've been looking at. And then in addition to that, because the supplement space is so underregulated, there's a tremendous amount of fake products. So on top of that, you have to fight that. So just to give you an idea in terms of the quality confidence, there are really only 2 reputable certifiers that will guarantee that the stuff inside is what it is. US Pharmacopoeia is one of them, and the brand called Nature's Made has used that a lot. NSF is the other. So for example, this is our company's brand, and we did invest in the NSF certified. But the problem, as I mentioned, one of the constituents, active hexose-correlated compound, was confused in early papers where people equated that with AHCC. So as a result, if you go on Amazon, even though people can violate the trademark, they've launched this thing they call active hexose-correlated compound, which has actually never been separated, never been sold separately. And then they describe it as consisting of a number of different and various mushrooms, which is not what it is. Whereas the genuine product you see at the bottom here says shiitake mushroom AHCC is extracted from the mycelium, the shiitake mushroom only. So again, you have genuine products on Amazon. Next time you have fake products, and the FDA enforces little of it. We're trying to get Amazon to help, but it's too complicated for them as well. So back to the science. So again, no such thing as alternative medicine. We need to focus on proven medicine. And there's a big contingent of kind of herbal history and herbal medicine and evidence-based medicine, and I hold a lot of respect for that. But in my experience, when we're trying to target the medical community, we really need to focus on the gold standard of medical science, which is ultimately the double-blind placebo-controlled trial with a statistically significant result. And as you'll see Judith present, this is exactly the protocol that she followed. And the only thing is that, unfortunately, given the regulatory environment, you'll have to settle for smaller trials, but they should still meet the gold standard and be statistically significant. So I hope that you all keep it in mind as you hear Judith's presentation. And on this note, I want to go ahead and introduce our next speaker, who will introduce our sort of feature presentation, which is Judith. And that is Dr. Gary Dang. I've known Gary for many years because he's sort of the legend in the integrated oncology community. He is the medical director of the MSK Cancer Center and a professor of clinical medicine at Weill Cornell. He really led and started the program here since 2003 and has been able to really help a tremendous number of patients. In terms of his background, he received his medical degree at Peking University Health Science Center and pursued a PhD in microbiology immunology at the University of Miami. He then did his clinical training at UT in Houston, where Judith now hails from. And then he did further research training at MD Anderson, where Judith used to be at. And he used to be the president of the Society of Integrative Oncology. I had an opportunity to spend some time in Japan with Gary, where he was the keynote speaker at a very large conference for nutrition integrative medicine, where he actually got to know AHCC. But most excitingly, when I thought like, hey, it would be amazing to have him present, I Googled him to try to see what he's up to. And I discovered this book that was incredibly well reviewed. So while we were all, I think, hiding out during COVID, although maybe it was written before, Gary managed to write this fantastic book called The Wellness Principles. And so I encourage all of you to pick it up. He promised to autograph it for me. So with no further ado, let me go ahead and introduce Dr. Gary Dang.
Thank you, Dan. It's really my pleasure to be here today to meet with all of you, and also have the privilege to introduce Dr. Judith Smith. I met Judith, how many years ago? I forgot. A number of years ago in a large international congress in Sapporo, Japan, where we talk about integrative medicine and what patients are doing, all this natural health products research. And I was very impressed by Judith Smith because there are very few people in the world that's dedicated to study these natural health products in such a rigorous manner. Because a lot of times these things are patients just take it and without knowing what they're taking. And because of this problem, Sloan Kettering Cancer Center established the Integrative Medicine Service 22 years ago, mainly to address patients' needs in this area. Because patients want to do other things other than just getting chemo and surgery. They want to do things they can do. So I just quote Michael Douglas, who's treated here. He said, "Cancer did not bring me to my knees. It brings me to my feet. I want to do something." Yeah. And a lot of patients, they go out, they try to change their diet, reduce their stress, exercise more, and they also take natural health products. And I have a PhD in immunology, so I know a lot of these health products do influence people's immune system. if nothing else, COVID taught us that, right? The same virus in different people can either give you mild or zero symptoms or very severe life-threatening symptoms. What are the differences? The constituents of the host, the immune system, the circulation, all that. And we see cancer sit in the same way. Cancer do not live in isolation. It lives in an ecosystem, which is our body. How we make our body stronger, the immune system stronger, less inflammation, less angiogenesis. Right now, there's no one single pill can do that. It is a lifestyle changes, including proper diet. That's why, inspired by my patients, I wrote this book. And stress management is also very important. What we do. And how do we get interested in this AHCC product? Because I love shiitake mushroom. If you are vegan, you know it's very hard to generate good flavor, the umami from a vegan diet. There are several secrets. One is tomato. It's my Italian tomato sauce. The other one is shiitake mushroom. All these mushrooms, they will generate this taste. So I was initially interested in that. I eat a lot of that. And then I run into these studies. I said, well, I know they have polysaccharide, they have all these immune stimulating properties, but exactly what are the effects? And that's why I came across through these studies, and also the companies make this product because I was impressed both by Judith's rigorous scientific research design, but also the companies, they really put their thoughts in how to generate and produce a consistent product. You know, that's a problem we have. We study a lot of curcumin, turmeric, and all the other green tea extract. If you come from different companies, the active ingredients totally different. We do a lot of chemical research ourselves. We run HPLC, they're all over the place. So I think this is a very good example of how to properly study a natural product with a verifiable, reproducible clinical outcome that can actually change the life of a lot of people who, in this case, would be HPV patients. So it's really my pleasure to have this opportunity. And let me just go through Judy's credential, which is very impressive. She is a professor and the director of the WHIM research program at the Department of OBGYN and Reproductive Sciences at UT McGovern Medical School, and also an oncology clinical pharmacy specialist at the UTHealth Memorial Hermann Cancer Center and the Children's Memorial Hermann Hospital. I did my residency there, so I still have a lot of memory. But I think our paths didn't cross because I have the place in she things. But it's great to see someone from Houston, and also later she became a faculty member at the Department of GYN Oncology at MD Anderson Cancer Center, and also provided the pharmacy service to the program. Dr. Smith is currently serving as the inaugural chair of the NRG Oncology Pharmacy Subcommittee. That she was instrumental in making it come into fruition. She's also board certified in oncology pharmacy, and certified professional in healthcare quality. Please, Judith.
And I should say, Judith's father art is in the audience, so give us a hand here.
First, I'd like to thank Dan and Marcia and Lillian for the opportunity to present to you all tonight. This has been a long journey, as both Gary and Dan alluded to. Started, I think, back in 2003 when I first was introduced to AHCC. Initially from the oncology, pharmacology perspective of can we give this to our patients? And Dan is 100% correct, and as well as Gary. So I took the approach of evaluating HCC as I do with the other INDs that we work on in our lab for various pharmaceutical industries and small startups. That's what our lab pharmacology side works on. So I took the same approach with HCC because I felt for me to share it with my medical community, it was going to have to pass all the same tests that we would with a drug, even though it is a nutritional supplement. And yes, there have been challenges along that way. So today we're going to focus in on the phase 2 study, but I'm going to touch on some of the journey that we've been. So I think this crowd probably doesn't need an introduction, but just for some of our colleagues and friends that may be online, just to define, HPV is a double-stranded virus that infects both the epithelial and mucosal surfaces of skins. It was first discovered in 1984. Some of us may remember it. I was still in high school, but once I learned about HPV, it's been my life career journey in focusing on it. It is a very common virus, and over 200 subtypes have been identified, but only 100 of those affect humans. One of the most common that we probably don't think of as HPV is plantarwarts. Very, very common. It was actually from our first pilot study, the most exciting finding for one of our participants was that for HPV, plantar's wart was finally eliminated after years of struggling with it. We are excited she eliminated our high-risk HPV. She was excited plantar's wart was gone. So it's a win-win for all of us. We really did focus in on the 15 strains of HPV that are associated with being carcinogenic or causing cancer. And I think what's really important for people to remember that HPV by itself does not cause cancer, but it's a cofactor in the equation, like Dr. Dang alluded to, that it's our immune system and it's something that can additional insults to the immune system that allow HPV to become and start the carcinogenic pathway. So it's estimated over 80% of women, and I always alluding and focusing on women because that's what I do. A lot of this extrapolates to all the men out there as well. But 80% of sexually active women will become exposed to HPV at least once in their lifetime. Highlighted in red here are the most common strains here in the United States, but when you look worldwide, those strains of HPV can vary, and that's why you see a variety of HPV strains included in the HPV vaccine. Just looking very briefly at how HPV infection occurs, it really wants to focus in on the E6 and E7 proteins that regulate viral replication. And as the HPV integrates into the cell, the E2 gene protein that normally downregulates E6 and E7 loses the tumor suppressor gene, primarily P53, which is something we really focus on in oncology, as well as retinoblastoma. And this ultimately can infect the cell and eventually lead to carcinogenic pathways. Important things, because I do get a lot of questions about this, low-risk HPV does not follow this pathway, and it's not a carcinogenic any of the data that we know to date. So why are people at risk of HPV? Definitely increased risk of exposure, which can vary from first activity. In this slide really focuses on intercourse, but we do know that any direct skin-to-skin contact, and that's really important for people to keep in mind that it doesn't necessarily have to involve intercourse. Multiple partners, it extrapolates and expands the number of people that are at risk. It is often a cofactor infection, so HPV usually travels in pairs with other sexually transmitted diseases. And one of our biggest challenge that we're really focusing in at UT McGovern is the decreased screening and reaching out to those low socioeconomic status patients. Patients have more access to healthcare. My ultimate goal is eventually to get to some of the third world countries like Africa, which I hope to do this summer, and summer of 2023, to really get this research to the patients that need it the most. Smoking can increase the risk of the carcinogenic pathway of HPV because of the active metabolite of nicotine, cocodamine, that is excreted in the cervical mucus. And then other immunosuppressive diseases and other oral contraceptives really gets a bad rap here, cervical cancer and HPV, because patients tend to not use the barrier method of contraception that will prevent transmission of the STDs, particularly HPV, but the virus itself is not estrogen. So as I said, this affects men just as well as women. About 1 in 4 men are currently infected with high-risk HPV in the United States, and 1 in 5 women are currently infected. So it's maybe even more of a problem for men if you want to think about it as we talk today, really more focusing on women. A lot of head and neck cancer, penile cancers, anal cancers are very prevalent as well. What can we do to prevent HPV? It's a topic for another day, but HPV vaccination is one of our only tools we have today. You can see here, this is a little outdated. The CDC is about to update this map, but in general, we're not doing great on the vaccination rates in the United States. We could do better here in the Northeast. Which I am originally from New York. I live in Texas now. I'm still a New Yorker. I still say I'm a New Yorker when I'm in Texas. I will always claim New York is home. But you can see we're doing much far better with vaccination rates up here in the Northeast and on the West Coast. A little not so much in between. One of the things that I have struggled from a scientific standpoint has been the ability to really hold true and not have whether or not the HPV vaccine is working as we move into clinical practice. Because during clinical trials, we can expose patients to HPV intentionally. That could be highly unethical. So they're designed to hopefully catch everything. But as we're learning and the data is growing, we need to learn more about how to optimize the use of the vaccine. We took a snapshot of 500 women in our women's health clinics in Houston, and only allowed patients that had another STD that they were infected with, and then looked to see whether or not they've been vaccinated or not. And they complete the vaccination series, which is really key. And this was just a snapshot. It's just a red flag and a signal that we need to look a little bit closer at this issue. Is basically the bottom line is we saw patients that were never vaccinated compared to those who had complete vaccination had the same rate of HPV infections they were looked at in this study. So it's just a snapshot, and as the data starts to continue to emerge, maybe we need to optimize the use of the vaccination, help the vaccine work better with supplements, or have a booster shot. So where and why HPV goes to cancer, you can see a lot of patients do get infected with HPV. Majority, over 70% will clear the infection in 6 to 18 months. So, and that is a good thing because otherwise we have much higher prevalence of cancer. But for those who have other insults to the immune system or not the ability to clear the infection and progress with screening, we can pick up these early stages and then prevent it going to cancer. So those, again, patients that don't have access to that screening, higher risks of developing into cancer. Ultimately, once it gets to CIN3, if not intervened on, it will inevitably lead to cervical cancer or related cancer elsewhere in the body. So when I started my research on HPV back in 2003, all we were talking about was cervical cancer. It was cervical cancer and HPV. I still remember sitting in the ASCO and the ACR meetings that I can't remember what year it was, but it was probably 2008, 2009, where we really started to say, wait a second, HPV is just as much a problem in neck cancer. And then it was probably another 4 or 5 years, we're like, wait a second, HPV is also really prevalent. And so you can see we now know both cervical cancer, anal cancer, head and neck cancers, both vaginal, vulvar, and then penile cancers were one of probably the more recent ones that we've added to the list of associated with HPV. So as I mentioned, prevention by vaccination is really our main tool in medical practice today. Early detection by pap smears, and now the Anchor trial with doing anal smear testing as well for HPV, and dysplasia available, but there's really, other than topical treatments for low risk HPV, hasn't been a really reliable source for treatment for HPV infections. Which leads us to AHCC. I think everyone in this room probably knows a lot about AHCC. It is the main ingredient that we have really found to be active ingredient that makes it unique to other mushroom extracts is the acylated alpha-glucan. A lot of mushroom extracts are primarily beta-glucans, which are immune stimulants. AHCC is more of an immune modulator, and these immune modulating benefits have been shown across a number of different modalities that have need immune support to help clear the issues. So When we looked, as I mentioned, AHCC was originally, I was interested in how it could help improve the side effects of chemotherapy, reduce toxicity, improve quality of life. A number of preclinical as well as clinical studies have been done worldwide. There's close to over 60 clinical studies worldwide that have been done with AHCC, with doses running from about 1 gram up to 9 grams. But that was a dose finding study, but the average, most of these studies look at the 3 gram dose that we used. And that's what we proceeded with in our clinical studies. Before any clinical studies, one of the things that is really important for every drug, as well as supplements, in my opinion, is to make sure that there's no potential for drug-drug or drug-supplement interactions. Not only looking at the phase 1 metabolism, which most of us talk about, and it is required by the FDA to list in your package insert as far as cytochrome P450 drug interactions, as well as really focused in with nutritional supplements on the phase 2 metabolism pathways. This is a summary slide. AHCC is a substrate of the 2D6 pathway. At high concentrations, we did see some induction of the 2D6 pathway. No clinical verification of this has been observed in the 100 or more patients that we have treated on studies. And then in the phase 2 metabolism pathway, again, some minor inhibition and induction on the UGT pathway. Again, this is not translated into any drug interactions observed in patients. Today, I can't go through all the bench to bedside paper. We'd be here for over an hour, but this is an open access publication in Frontiers in Oncology. It's available on HCC research website if you want to go pull the link for it. But we did take a very systematic approach. We started in the bench in cervical cancer cell lines, moved to animal studies. I have an amazing team that stood up with my need for perfection and details and repeating things. I think we did animal studies 3 or 4 times before I had any real faith and the results were reproducible. I think I've been my own hardest critic of this research over the years. And then that led to our pilot studies. Once we really saw some activity in the mice, that was a durable response. We did 2 pilot studies. And that was when we first shared that data at the Society of Integrative Oncology meeting in 2014, that we really thought we might have something that may be beneficial. And then that data from the 2 pilot studies was used to fund our NIH grant that led to the phase 2 study. So this is just the citation for the phase 2 study we'll be talking about today. That AHCC supplementation to support immune function to clear persistent human papillomavirus infections. So this was a phase 2 randomized double-blind placebo-controlled study. Again, trying to mimic the same approach that we would take with drug development. It is required to do double-blind and placebo-controlled. Patients can't control their HPV status. We did want to make sure that we were blinded to the results. We were looking at toxicity in the study, which is why the investigators needed to be blinded as well as the patients. And then we wanted to follow it up for durability of response. So this study evaluated the efficacy of HCC 3 grams. We had previously looked at the 1 gram dose that in our pilot studies, it was effective, but it just took longer to reach the same goal. So we did proceed with the 3 gram dose. It's taken once daily on an empty stomach to eradicate HPV infections. And these women had to have HPV positive pap smears. I'll go through the eligibility here in a moment. We wanted to look at the durability response, which was most important to us, and then compare and contrast AHCC to placebo as far as adverse effects. The one thing, there's a lot of information on here, but I want you to focus in on the inclusion criteria. When we initiated this research, HPV was typically focused on the risk of persistent infection in women over the age of 30. It's now 25. So on our next study, we did lower the age to 25. But in this when we initiated these studies, we stuck with 30. Women had to have positive HPV pap smears for over 2 years consistently. So it wasn't just one pap smear test. We had to have evidence and data to support that they had persistent infections. Majority of the patients that did enroll in the study had greater than 3 to 5 years, and some up to a decade of being positive. We also did make sure that they were not pregnant, since we are still looking into the safety of HCC during pregnancy. They were excluded during this study. Otherwise healthy. We didn't have any patients with active cancer because we did not want to interfere with their treatment. And then we did exclude taking any other nutritional supplements because we did not want to interfere with potential observations with HCC. So this is a schematic summary of how the patients were randomized in the study once they were enrolled. They were either randomized to group 1 that received HCC for 6 months, followed by 6 months of placebo. And then, or they were randomized to group 2 that got 12 months of placebo. At 12 months, the patients were unblinded as well as us. We looked at their HPV status. For the patients on the active arm, if they're HPV negative, we continue to observe them for another 6 months. If they were HPV positive, they were considered a non-responder, taken off. On the placebo arm, if they were HPV negative, they were taken off study as not being positive or negative anymore. There was a placebo effect. Or if they're HPV positive, we did offer them at that time to receive HCC unblinded for 6 months, and then continue to follow them 6 months off of the HCC as well. And that was done intentionally to incentivize. HCC is readily available on the market today, so we're trying to incentivize patients to enroll in a study where they could just go out and buy and take it on their own. So we're trying to help with enrollment and accrual, but that you'll see as we look at the results, we had a little bit of a challenge on that part. So one of the other key things while we were looking at this research is right around 2015, there was the introduction and market of the mRNA HPV assay, which picks up active HPV infections. And our first couple of patients that came to enroll, all of a sudden they were negative. We're like, oh, wow, good for you. The first patient, I was happy. It's like, good for you. That's great. Decade of being positive. Now you're negative. That's awesome. Second patient, I was like, wow. But the third patient, I was sitting in the pathologist's office and what are you doing? What's going on? This is impossible that all these patients, like all 3 of them have been positive for over a decade, all of a sudden are negative. And that's when we had a big discussion about the role of mRNA assay being much more sensitive of picking up early cancer changes in the cells versus what we were looking for, detection and prevention of HPV in that dormant inactive infection. So had a student go back, look at 22,000 pathology reports. That's the great part about being in medical school is you can delegate these things. So, you know, she was very interested in trying to figure this out. At the end of the day, as my gynecologists and I like to joke, is that we could guess on HPV status better than the mRNA test, because about 70% of the time, patients were telling they're HPV negative and they were HPV DNA positive. So we brought this forward, and the International Pathology Association was aware and said that clinicians should know what they're testing and should know whether or not they should be using an mRNA or DNA test. Not sure the clinical clinicians out there all were told that they should be knowing. We've tried to promote this and educate. Some of the guidelines are now specifying HPV DNA status, but not all of them. But we continue to try and educate on this. And this was published in WHIM and was the editor's choice for that issue. That's trying to make sure people are aware. So on our study, we decided to test both to continue to validate this data in real time. So both RNA testing as well as DNA testing next door at our sister institution in Methodist helped us completing the DNA test. We also then looked at the immune panel for validating HCC, this mechanism of action. This is kind of shows you where our patients were enrolled, and just to give you the exact numbers. So the eligibility, we had screened over 144 patients. 7 patients were excluded for either not meeting inclusion criteria or declining to participate. Of those that were randomized, in the placebo arm, we only did have 19 that finished the study. That was partly to do with that patients were able to go to their own gynecologist and find out their HPV status. And once they realized after 6 months that they were still HPV positive, went off study themselves because they were not on active. Frustrating, but part of the research. Majority of the patients on the active arm did continue on. So this is a summary of our demographics. So you can see patients were about the same age. Unfortunately, it's been a challenge for all of us in research to get diversity in enrollments. We did have a primary Caucasian patient population. We hope in our next study to get some diversity by reaching out to some other areas in Texas for collaboration. And majority of the patients on the study had a similar number of previous sexual partners, and everyone was either not sexually active or in a non-close relationship during the study. This is where it gets exciting when you look at our results from our patients that completed the study. We had 14 patients, or 63%, that were both HPV negative, both by RNA and DNA after 6 months of AHCC. Of those 14 patients, 9 patients were still negative 12 months after stopping AHCC, so they had a durable response. There were 2 patients after 12 months that were HPV negative by the RNA, or so they didn't have an active infection, but they did become DNA positive, or were continuing to be DNA positive, showing a partial response. And then 3 patients that were both HPV, RNA, and DNA positive 12 months after stopping HCC. And then we had 8 patients that were non-responders. In the placebo arm, it's always really exciting when you're a researcher and your power calculations come out exactly as you estimated. We estimated a 10% placebo response, and that's exactly what we saw. So it was super exciting that we reached our power calculation. Because this study has not, nobody had ever actually tried to do this before, so we're totally estimating that based on clearance of HPV. So we had 17 patients that were positive at the conclusion of the study. Of those that were positive, 12 continued on and elected. A lot of patients were coming coast to coast, traveling to Houston for the study, so they were like, yeah, I'll just go buy it and continue to buy a plane ticket to come here just to get an HCC. So we helped reach and get the HCC supplement off study, but of the 12 that continued, 6 had a response. They were both RNA and DNA negative at the conclusion of HCC with a durable response at 12 months out. This is one of our mechanism studies that I really want to walk you through. It's important to make sure everyone's aware that interferon beta is a type 1 interferon, and it's associated with the virulence of a viral infection. So the higher level of interferon beta is, the more likely you have chronic viral infection. This is similar for HPV, CMV, HSV. And so these high levels of interferon beta will actually suppress your type 2 interferon, which is like your interferon gamma, your NK and T cytotoxic cells that are necessary for the immune system to come in and clear that viral infection. So very nicely at this very nicely at the same time that we were doing in the pilot study, when we first evaluated this mechanism, the knockout models have already established the same mechanism with CMV. So we didn't go back to the animal lab to confirm it again with knockouts. But on this purple line at the top represents the placebo group. You can see that the interferon beta was elevated and stayed. I'm sorry, not this is the non-responding group. I apologize for that. This blue line is our placebo group that at 12 months started the HCC of those 12 patients. You can see their interferon beta dropped nicely. This light green line over here, this is our complete responders. You can see at 6 months, their interferon beta level had dropped very nicely, and they had a durable response and did not elevate again for the rest of the study. this is what was really interesting, where we learned the most, that led to our current R01 application, where we saw the interferon beta level came down, and that's why we saw HPV suppression, where both RNA and DNA were not detected at 6 months. However, this was placebo and blinded, so we didn't know. They were stopped, the AHCC was stopped, and then you can see it comes right back up. So that's where we learned from there that we have to monitor both interferon beta and HPV status, and that's what we'll be doing in our next phase 2 study. When we look at the T cell response, again, trying to keep the color schematics the same as the previous slide. The green represents the complete responders, red are partial responders, purple is our non-responders, and blue is the placebo group. And you can see in the green and red at 6 months, and a very nice T cell response. It stays durable throughout for the duration of the study for the complete responders, and starts to drop off for the partial responders. We also looked at the interferon gamma that helps clear with that type 2 interferon and clearing viral infections. And again, we saw a similar pattern where at 6 months, both the complete responders and partial responders had a very nice response. It was a much slower decline for the patients that had complete response, and more significant in those partial responders, as well as the placebo group had no change. A lot of the AHCC literature, primarily in oncology, really looks in at the mechanism of action on NK cells. We're not sure, because NK cells may not really be a big part of HPV infections. This is probably more related to cancer. We weren't able to reproduce that in our study. This may be more appropriate to look in oncology studies, and we were just trying to check all the boxes of previous mechanisms that have been proposed, but NK cells are not playing a big part in viral infections. As far as side effects, this was really exciting, as we had basically compared to placebo. AHCC was very well tolerated, really no different. We saw nothing more than a grade 1 toxicity, which usually resolved shortly within the first 2 to 3 weeks of taking something. So the conclusions from this study is, this phase 2 study demonstrated the AHCC supplementation was effective in modulating the immune system to support complete eradication of high-risk HPV infections. Our response rate around 63% is consistent with both our pilot studies that were for smaller studies, but our response rate seems to be fairly consistent, and what we've learned from that and moving forward, and how to optimize that. AHCC was extremely well tolerated. We're very excited and focusing in on how interferon beta could be a reliable biomarker for monitoring for not only response and clearance of HPV, but perhaps maybe even earlier detection for both men and women. Our future research is really going to be focusing in on that duration. Kind of 20/20 hindsight is that everyone's immune system is different. So stopping at 6 months was probably premature for some patients, and we now know that we monitor both interferon beta and HPV status, we might be able to define that duration. I'd like to acknowledge a lot of contributors. AminoUp was very, very supportive in the initiation of our preclinical studies. After we moved beyond preclinical studies, we then moved to support from my chair in the Larry Gilstrap Perinatal Women's Health Research Fund, and help fund some of the pilot studies, and then some other philanthropic funds, and then our phase 2 study was funded by NIH. I'm very grateful to my research team in the lab, as well as my collaborators. I don't know if we have time for questions.
We do.
Questions?
There's a couple of the chat. Yeah.
It's just
Comments?
yeah, no.
No question.
I have a question. Because we've gone on about this for a long time. I have never heard of this, the DNA test. So can you explain the difference of the two tests to me?
Yes.
Anybody else who's not aware?
So, the mRNA, they're both done the same way, so you would not know unless you know what goes being ordered. So the collection is the same, but the DNA test is much more labor intensive, where the sample is processed, and it's looking at the PCR test, where it's the RNA test is a high throughput assay, where they can have earlier detection of those cellular changes. It's much more sensitive. It's high throughput, so that's why majority of the labs, Quest and LabCorp, and a lot of institutional labs have switched over to the RNA test. And so unless the clinician specifies they want a DNA test, they will get the RNA results, which is concerning from a clinical standpoint because people are being told they're negative when they're still positive. So that's why we overly emphasize to make sure, like when patients email me directly, they're like, "Oh, I'm so excited, I'm negative." Again, I'm my own worst critic. I'm like, "Did you make sure it was DNA?" Like, because you may not be. And they're like, "What?" Like, I mean, I can take it and run with it. They're like, "Oh yes, everyone's being negative," but it's really, really important to make sure you're getting DNA testing.
Well, that seems to me like that's a very big deal because we're in the know and I've never heard of that. So has anybody else, doctors, people here heard of this? And you test patients and tell them they're clear and don't, and you know which test is being done or?
So
Okay, so
we have a long, we presented at a lot of OB-GYN meetings, and it's the first that even my OB-GYNs have heard about it. We obviously had a big education campaign at UT because it really directly related our enrollment on the study because of what's going on. So it's really important. SGO, which is the Society of Gynecological Oncology, is aware. ACOG is aware. We're working on trying to get better awareness, but that's anything else people take home online or today is make sure you're asking for HPV DNA testing.
Is the RNA a thin prep?
They're collected, but they're all thin prep collections. There's no difference in the collection, it's how they're processed.
But the DNA, is that the co-test?
No, their HPV testing is a co-test from pathology and DNA. It's what they look.
So if someone asks their gynecologist, can you give me the HPV DNA test? So they
have to say no. A gynecologist, like a lot of my patients would be like, "What do you mean?" And I send them this paper, and then they go and so you have to write it specifically. We write it specifically on our lab. The pathologists get annoyed with us because the manual processing versus high throughput.
Would you say that most gynecologists then are unaware? Like, are we talking about something that you know about, but
it's a big gap in knowledge that we're trying to.
Sorry to focus on that.
No,
It's
great.
great,
It is
but-
very important because we would do a lot better because it's a little bit frustrating when patients think they're negative because what that means from a screening perspective, if you're negative, you don't have to come back for 3 to 5 years, depending on which guideline version you're following. And in that 3 to 5 years, if you have immunosuppression, you have a stressful event, you have immune deficiency, or anything else that could trigger that carcinogenic pathway, versus being, if you're HPV positive, you're screened at minimum annually, if not 6 months.
Dan, we have more work for you.
Judith, this is not directly related to the study, but you had mentioned to me at lunch about the vaccine and how long you think it lasts, and also at what ages they should be recommended. I was curious. This relates to our other work, obviously.
So the HPV vaccine definitely has helped us so much in this arena. I think we just need to continue our work. I think we got excited and everybody just ran with the first recommendations and vaccinating at a very young age. We do know that it's for the 9-valent vaccine that's currently available. The latest estimate is 7 to 9 years. So you need to revaccinate a booster, and those booster recommendations are not been established. So personally, my clinical opinion is to vaccinate a little bit later, maybe not at 9, but maybe closer to the teenage years so that it gets them at least to college, but maybe that we really need work on that boosters where I really like to see the research focus in on. And then, as I mentioned at lunch, just trying to figure out a way to make the vaccine work better. We have data with AHCC improves response to the flu vaccine. Maybe it could do the same with HPV vaccine.
I had a question or maybe a concern. When you talked about like the on the slide about people more susceptible to getting HPV, and it was multiple partners starting intercourse younger. And then you finished by saying, but actually this is contagious by skin to skin contact. And I just wonder if for the entire world, if it wouldn't be good if that were in black and white, because what you have are the middle of the country going, my child won't have sex, da, 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 da. But if you put the skin to skin contact up there, then I think it will help understand, get people to understand the virus part.
No, that's a very good point. It's
Just a
definitely
thought.
any direct. It's more intimate skin contact, other than like from shaking hands.
Right,
It's
but
not
you
from
could say-
casual contact, it's more the intimate skin to skin contact. That isn't very important.
I just feel like in terms of getting that, like the rest of the middle of the country, anytime you can sort of not make it seem like, oh, those are just such big risk factors, like my kid would never,
No, that's a very good point, definitely.
right? So
Update that, I do a lot of community education, so I will try.
just to help our cause.
No, absolutely. Great point.
One big point of debate, I wasn't gonna go there, but we might as well, was, and I think we
Because she looked at me.
exactly. And I think you went back and forth a little bit with Dr. Posner is whether or not you can fully clear the infection or whether you clear an active infection. So his view is you can clear an active infection that becomes dormant, which has a lot of value in itself. And then obviously you provide a different perspective. I think our conclusion is it doesn't matter because even if you clear an active infection as dormant, you're still achieving significant benefit. But since you're on camera anyway, I thought maybe you could share with us like why your thought is different.
Thank you. That's like, it is a big area of controversy in the whole HPV community. In our studies and our research, we've been have patients on studies now since 2013 that we still are following some of those pilot patients that are still remaining HPV negative. We had a couple of our pilot patients who should make it clear now is AHCC does not prevent reinfection. And our pilot patients did learn that lesson the hard way. And we were able, in N of 2, show that restarting AHCC did clear it the second time they had it follow them. So I think, if you look at the portion of patient population, 80 to 70% of us become infected with HPV, that the prevalence of HPV related cancers would be much higher if we could never clear it. Because especially here in US where we have high stress lives, I think people have sleep deprivation, eat poorly, don't exercise enough, and have tons of high stress jobs across the country. So I think rates of HPV related cancers would be much higher. So I do think if you can, intervene with HCC, or majority of patients do clear it on their own first place. I think that it is possible, but yes. I think a lot of the controversy comes up with the mRNA and DNA tests has led a big crux to this argument because patients are told they're negative and they're mRNA negative. And then it comes back positive a couple years later. Well, they were never negative in the first place. They were DNA positive the whole time. So I think that has confused some people in this arena, like, oh, yeah, it does come back. You never get rid of it, but they're testing RNA, not DNA. But if you can truly establish DNA negativity, it's different.
Because sometimes it never showed up and then all of a sudden it shows up. So you always had it, right? But it's just showing up, no?
No, because the testing is, the sample testing, that's actually what the pathologist had us chasing down a rabbit hole with our first 3 patients that were coming back saying, oh, you need better sampling. And so we really explored that. So there is poor sampling, so there's false negatives, but not necessarily. In this study, we were actually using 3 modes of sample collection all the way through, because once we started it, I didn't want to go back to the IRB and say, oh, well, actually we figured out the assay was the problem, not how we were sampling it. That was too confusing. So we collected samples 3 ways. Yes.
Judith, you said that, I think you said that you're gonna do another phase 2.
Mhm.
From a layman's perspective, what is the goal of the next phase 2? Does it elevate the status of AHCC? You know,
So
Doesn't
what
need to
we're
be
really
phase 3.
focusing on are just partial responders, the people that we stopped. And we are going to be testing both the interferon beta status as well as HPV status, and not stopping them until they're both have an HPV negative DNA test as well as interferon beta below 20, and then treating them 3 months longer and then stopping it. It's just that durability of response and figuring out the duration and a way for clinicians to be able to personalize how long someone needs supplementation.
There may be a question in the chat.
Excellent presentation.
Thank you.
Thanks, Judith. I don't know what to say, but that was heavy. Awesome. Thank you to the online group. Thank you very much. Thanks to all of you. And I also want to acknowledge 2 people here. One is Dr. Paul Romasak for helping us with the room. Thank you so much. Thank you. And also to Amna. She's our intern. She helped us with everything, especially the name tags is very nice. So thank you so much. And of course, thank you to everyone, especially Dan and Marsha and Maggie and Danielle, just everyone. Thanks so much for being here. And now let's mojo.
Sure.